I'm Leila Varghese. I'm a postdoctoral fellow at the Ludwig Institute and the Ludwig Institute in Brussels, Belgium. So I'm going to talk today about how a variant in CSF2RB drives leukemogenesis in patients with Down syndrome. So next-gen sequencing has really opened up a whole new world for researchers. Now we have such a great idea of all the variants that can occur in patients. But the question really is, well, how do these variants drive disease, and do they drive disease? So we were interested in patients with Down syndrome who develop myeloid leukemia. So these patients are at a much higher risk of developing myeloid leukemia, and it's preceded by a unique pre-leukemic stage called transient abnormal myelopoiesis. Sorry, that's quite a mouthful, but we call it TAM for short. And anyway, our colleagues recently did a study of the largest cohort of these patients and did next-gen sequencing, were able to find a whole lot of new variants that occur in either the transient stage or in the chronic stage, which develops later. So we were interested in one particular mutation which came out, which was in CSF2RB, which is a cytokine receptor, which signals through the interleukin-3, interleukin-5 receptors, as well as the CSF receptors. And our colleagues were able to show that when you put this variant into cell lines, it can cause proliferation without any stimulation. But it didn't do that in all cell lines. So to us, we wanted to really understand what the mechanism that was driving this. So normally this receptor requires a second chain to signal. So it's the transmembrane receptor. It sits on a surface cell and it forms multimers with other cytokine receptors, these alpha chains, so either the IL-3, IL-5 receptors or the GM-CSF receptors. And we thought that perhaps it was the expression of the particular receptor chain that goes with CSF2RB that might control whether or not this causes proliferation in different cell lines. So considering it didn't work in all cell lines, it didn't cause this abnormal proliferation, we thought that it's not just dimerizing with itself, it requires another factor. So we tested a few different receptors, and it turns out that thrombopoietin receptor is what's needed for this cytokine-free proliferation to occur, so to drive the cancer. And we worked out a few elements that were necessary for the receptor, and we think overall that it probably dimerizes with a thrombopoietin receptor and sends a signal that would normally occur only when thrombopoietin is stimulated by thrombopoietin receptor is stimulated by thrombopoietin. And this is kind of interesting because we've never seen this kind of interaction before between these two receptors, and probably gives us hints that maybe even in adult AML, we might want to also look for mutations in CSF2RB.